Hello everyone, thank you very much for joining us for our talk today by Professor Timms from Australia. The talk title is Developing Vaccines Against the Two Major Infectious Diseases Threatening Koalas, Chlamydia and Koala Retrovirus. Thank you very much, Professor Timms, for joining us, even though it's nighttime in Australia, despite the huge time difference. I shall start with the introduction for Professor Timms. Professor Timms is Professor of Microbiology at the University of Sunshine Coast in Queensland, Australia. He is a nationally and internationally renowned microbiologist with specific expertise in the area of Chlamydia and Koala retrovirus. His laboratory is acknowledged as the leading Australian laboratory and one of the leading groups internationally working on all aspects of chlamydial infections. His research group is developing vaccines and new diagnostics for chlamydial diseases in humans and animals, as well as an improved understanding of chlamydial genomics, cell biology, and pathogenicity. He has published over 280 papers, reviews, and book chapters in peer-reviewed international scientific journals. In terms of the structure of the session today, the talk would be around 40 minutes-ish, followed by a Q&A session, which will be moderated by Christoph, our co-president. Without further ado, let's welcome Professor Timms to deliver his talk. Thank you.
Yes, well, thank you very much for that introduction, and I'm pleased to be here to talk tonight, or the morning as it is there, to hopefully give you some insights into koala diseases and what we're doing to try and develop vaccines. I'll just get ready to share the screen for everybody. I think that's right. Just trying to make sure that. Okay, hopefully that works for everybody. Is it, just confirm that? Yeah, I think that's okay. So if that's okay, I'll get started. So yep, again, thank you. Okay, that's great, great. Yeah, it's technology is an amazing thing. So I'm going to talk to you about mainly about chlamydia, because chlamydia is the major disease affecting koalas, but also our work with the koala retrovirus. It's a new challenge. And together, these are the two most important diseases. So I always like to start these talks acknowledging that there are a number of factors that are really threatening koalas. In the top left-hand corner, I'm not sure how easily the mouse pointer is to see, hopefully okay. Obviously, habitat destruction still is the major issue, but also over in the right-hand corner up in the top, this is an unfortunate animal that's had an interaction with a dog, sometimes domestic dogs or wild dogs. Down the bottom left-hand corner, koalas crossing roads. So clearly roads are a major issue. And indeed, most recently, climate change in the form of bushfires, wildfires, and heat stress. So all these things are clearly very important. But on top of that, the major non-human factor is chlamydial disease. And as I said, I'll mention koala retrovirus as well at some point. Just checking the time. So I'll point out to you some of the diseases chlamydia causes to begin with. So chlamydia causes two major diseases in koalas. It infects females and males, but in females it's the most important because it affects their reproductive tract. So on the left-hand top corner here, it causes infections in the urinary tract, so it results in urinary incontinence, which results in urine and wetted koala fur, and sometimes the term wet bottom is coined. But as well as that, it ascends the urogenital tract and it affects the upper reproductive organs. And in doing so, it causes blockages, which result in infertility. But as well as that, you can see on this right-hand panel up the top here, it also causes these large cysts. And these cysts are not only medically important for blocking the tubes, but they also are very painful for the animal. So the reproductive tract disease is clearly the most important. But in addition to that, it also causes ocular disease and infects the eyes. And indeed, these are the two very parallel things to what Chlamydia trachomatis causes in humans. It causes urogenital tract disease and ocular disease. So in the koala here, in the middle at the bottom, you can see a koala where it's got one eye severely infected here and the other eye very little infected. So it causes Starts out with conjunctivitis, which might be mild swelling, and eventually, as with time, it closes over, and because the eyelids get scarred, scar tissue forms. This is similar to what happens in humans with trachoma. So these are the two major diseases. On their own, they may kill some koalas, but they certainly make koalas difficult for them to feed and to survive. So in humans, of course, we have antibiotics that are very useful for treating chlamydia. But there are three main reasons why antibiotics are not as useful in koalas as a treatment priority. The first one is down the bottom corner. It's meant to point out the liver. So koalas have a very active liver with lots of liver enzymes. And the reason they have this is because, of course, they're eating eucalyptus leaves all the time. And these gum leaves are full of toxic components. So the liver has expanded its P450 enzyme repertoire to digest these toxic compounds. And therefore, when we give them antibiotics, they digest those as well. And normally in humans, you might give antibiotics for maybe one or a short course of antibiotics. In koalas, we have to give antibiotics daily for 14 days and often 28 days. So that's a problem because it's hard to do that just to get overcome it. But the problem happens in the panel above, in that at the same time, these antibiotics for 14 or 28 days are killing the chlamydia, they're also killing the gut microbiota. So the koala, like us, have lots of microorganisms in the gut. Of course, in the case of the koala, this is extremely important because these microorganisms are the ones that digest the eucalyptus leaves. So what's happening is at the same time we're using antibiotics to kill the chlamydia, we're also killing off the bacteria that would otherwise be used to digest eucalyptus leaves. So in many cases, treating koalas with chlamydial disease with antibiotics, often in at least 50% of the cases, has an extremely bad outcome. So these are the two main reasons why treating with antibiotics is a difficult situation in koalas. So as a result of that, one of the options for ensuring that chlamydial disease is not as important as it might otherwise be is development of vaccines. So we've been trying to develop a vaccine for 10 years now. I'm not going to talk to you too much about the animal model of the mouse, but of course a lot of our information has come originally from the mouse. But our focus has been for the last 10 years focusing entirely on the koala. So just a little bit of taxonomy for chlamydia. Nothing overly important, except the species of chlamydia we're talking about is Chlamydia pecorum. It infects a range of hosts, but it's a dominant organism in koalas that causes, we think, the majority of infections and the majority of disease. So this map across Australia, it is here, is meant to show you a little bit of epidemiology of where chlamydial infection as well as disease occurs. So the red bars in the pie charts indicate levels of infection. So you can see that chlamydial infection occurs across the majority of Australia. There are a couple of island colonies. Artificially, koalas have been put there. So in those particular cases, purely by chance, there's no chlamydia. But by and large, there's chlamydia. And so you can see, for instance, in where I live in southeast Queensland, chlamydial infection rates range from 30% up to 70%. So this means that many of these animals in these populations, 70% of koalas are infected with chlamydia. The numbers are lower down south, and there's different disease as well. So not all koalas that get infected have disease, but perhaps 50% of them that are infected end up with disease. So this is a major problem and a major threat to the long-term survival. So we set about making a vaccine. We've been doing it for a while, and it's not a single vaccine. As I just go through the different components here, you'll realize that they're actually up to 3 or even 4 different vaccines, and because they have different immunological requirements. So the first case, the sort of vaccine you might want is for a koala that has no chlamydia, so it's chlamydia negative, and you might want to vaccinate that animal so it doesn't get chlamydia. That's the hardest thing to do, and I'm not going to talk to you about that at all because it's very hard to measure it and it's very hard to do it. The second group, which you'll hear more of our vaccine studies, is in the case where the animals are chlamydia positive. So we use the PCR test routinely to detect chlamydia. So in this case, the animals might be PCR positive, so they're positive for chlamydia, and a vaccine we would hope would then reduce the level of chlamydia, either to a level that then wouldn't be allowing transmission from one animal to another, or the second thing we'd like it to do is if we could reduce the level of infection, that we could reduce or prevent even the infections going on to develop into disease. So there's 2 different sorts of components going on there. And then finally, and you can see it for this koala on the right-hand side, is animals that are already clinically diseased. Can we make a vaccine, a therapeutic vaccine against these koalas? I'm going to give you some good results there today, but 5 years ago, I would have said that's almost impossible because it's the immunopathological response that drives this disease in koalas. So if you think about it, it would be quite difficult for a vaccine to not only reverse infection, but to reverse the disease pathology. But nevertheless, that's also some data I'm going to show you today, and it's also looking quite promising. Because as I mentioned, anything up to half the koalas that are infected will go on to get disease. A little bit of immunology, not too much. So chlamydia is an intracellular parasite. So this is meant to represent the cell here. And so the chlamydial elementary body, which is floating around in the environment, infects the cell, gets inside, that it actually changes shape and conformation and becomes a reticulate body, and it grows exponentially to produce about 500 chlamydial particles per cell that this eventually usually lyses and spreads the chlamydia to the next infecting cell. And on the right-hand side here, I've just got an actual photograph under the microscope. The red is the cells, and the green coloring here is the fluorescently labeled chlamydial inclusion membrane body, which has up to 500 or more chlamydial particles in it. So there's two sorts of parts of the immune response we might want to use. We might want to develop antibodies that will bind to the chlamydia. And this is very similar to what we're going to do in the flu virus or even in coronavirus. It's quite topical now. And that's useful. But secondly, because it's an intracellular parasite, we're going to want to develop as well as strong cell-mediated immunity, a Th1 immunity. And indeed, in the chlamydial world, we know that both of these two are required. What I'm going to tell you, the data I'm going to show you, or the limited amount of this stuff will be antibodies are easy to measure in koalas. We don't have the same sort of resources to measure it in cell-mediated response. So what I'm going to do is I'm going to run through a couple of our field trials, summarizing the results that we've obtained over a number of years now, to focus on protection. There are two stages in making a vaccine, of course. One is to make the vaccine and get the immune response. And I can tell you that when we vaccinate koalas, we do get an immune response. We get an antibody response, and we get a cellular response. And the question is it the right response? Interestingly, in our world with coronavirus, it's exactly the situation where all the coronavirus vaccines are up to. Many of them can be used, they're safe. They vaccinate humans. You get an immune response, but the question is, does it protect? So this is the key thing. So that's what I'll focus on today. So what does the vaccine look like? It's got three components to it: the antigen, the adjuvant to enhance immunogenicity, and then the delivery mechanism. So the antigen we've been using is a major outer membrane protein. So this on the bottom right here is a diagram of the major outer membrane protein, a bit like the flu vaccine as well, same sort of thing. It's part of the protein is buried in the membrane, and part of it is sticking out the membrane. This is usually the part that's most immunogenic, but of course it's also the part that changes. And so the same way that the flu vaccine has different strains, and therefore each vaccine each year needs to be improved to make sure it works against the different strains or the different antigenic epitopes. In the case of our koala vaccine, this is the same thing as well. At the moment, we've got 3 particular genotypes of the Chlamydia PCR from koalas in our vaccine. So that's the antigen. Secondly, we use an adjuvant. I'm not going to talk too much about it, but you'll see in some of the studies when I mention them, there are 2 components, either an ISCA matrix adjuvant, which we normally have to give in 3 doses, and a bit confusing here, sorry, but this adjuvant here is the one we're using now. It's got 3 different components in it, but we only have to give it as a single dose. And obviously that's a big advantage for something like a wild animal like a koala. And finally, we're currently delivering it by a subcutaneous. We've tested lots of different means, intranasally, orally, and while they all might seem better or different, we've settled at this point in time on a subcutaneous delivery. So let me just, okay. So before I head off into it, let me just remind you how difficult and expensive it is to work with koalas. And as cute and as cuddly as they are, they're difficult animals to work with. So this is just in southeast Queensland. This is the process by which animals are removed in the wild. So this is a long, long ladder up a tree with a skilled person. You might be able to see the rope they've got, and there's a bag at the end, some sort of way, and they're trying to get, this is the koala right here. So they're trying to get this koala to move down the tree. So my point here is that every time we do a field trial, so we'll do some trials in captive animals, but every time we try to do a field trial measuring the real situation, it's extremely time consuming and it's extremely expensive. Some of the trials I'll show you, we've done them because we've matched up with other field groups doing them. If we had to fund them themselves, each trial can cost in the range of $1 million. So they're very expensive trials. So let me talk to you about one of the first ones. So we've done 10 trials now, or 10 different vaccine evaluations. This particular one I'll mention was where we had an opportunity because through the middle of these colored shapes here is a train line. So a new train line was being built. This was fortunate for us, or unfortunate perhaps. And the green parts, you can see parts of the train line were going through koala habitat. And this meant that the government was required to spend money to make sure that koalas were looked after during this process. And so an ecological team was responsible for capturing all the koalas, putting radio monitors on them, and following them almost weekly across a several year period. This meant that we had an opportunity to do a vaccine trial in these areas here with a lot of the cost being taken by somebody else. So this first one I'm going to do is it's going to highlight that vaccine reduces the level of chlamydial infection. As I said before, if we can reduce the level of infection, that's a pretty good step in the right direction. So I just put the mouse on the left-hand side here to remind you that in a mouse trial where all of this is the model that's being used for koala vaccines, in the mouse situation, we'd use female mice completely. We'd grow them from a single litter. They'd all be exactly the same age. They'd have the same genetics. We'd know when we vaccinated them. We'd take samples at known periods of time. We'd even use hormones to synchronize them. In the right-hand side, but when I'm going to tell you about our koala vaccine, of course, we don't have that same possibility. We'll be using male and female koalas. We'll be using them at different ages. They'll have different genetic backgrounds. We'll give them the vaccine and we'll capture them at a time that we're not sure about. So if you can see on the right-hand side here, this is at 0 time, and this is at 6 months later. So this is a pretty stringent test of any vaccine, much more stringent than you would do in a mouse trial. So what we have here is in unvaccinated animals, and these particular ones, this is at the urogenital site. So this is a logarithmic graph on this side. So this shows you at the beginning, this is the range of chlamydial infection as determined by PCR test. And then after 6 months in the unvaccinated group, there's a range because there's an animal here, but by and large, there's not much change in the level of infection in the unvaccinated group. By comparison, on the left-hand side now, this is our vaccinated animals. So these are about 30 animals per group. As I mentioned, 30 animals might or might not sound like a lot, but it costs us $1 million to do this trial. And so here hopefully I can convince you that what started here in the vaccinated group, and then this one animal is pushing that, but the mean is down here. So this is a pretty dramatic decrease in the infections in these vaccinated animals. Perhaps even more impressive is now at the eyes, at the ocular site. So here we've got the unvaccinated group going from the beginning to after vaccination. In fact, on average here, they've increased a bit because time of the year matters. Whereas again, here we have the eyes and the vaccinated animals having a very dramatic, and I think hopefully you can be convinced, pretty significant drop in infections in these two groups. So this is our first field trial. We've done 3 or 4 trials in captive animals up to that point, but captive animals, of course, don't usually have chlamydia. So this was our first test to see whether or not the vaccine gave some level of protection. So at this point, we were pretty happy, starting to put some pieces of the puzzle together and see how we go from there. So I'm just going to mention the next trial. Again, it's in the same group of koalas. There are about 300, for what is interesting for you, there are about 300 koalas in this corridor here. So in the first trial, we used 60 of them. Interestingly, when we came to the second trial, which is a year or 2 later, the infection levels were not as high. And we knew that would be the case because part of the veterinary monitoring of this was to remove those high infections. But nevertheless, at this point, we've gone across to our current version. It's got 3 antigens in it, and it's using our triadjuvant. So we only needed to give 1 dose. So I presented that somewhat differently here. So in the groups of koalas, the 20 or so in this group here, a lot of them didn't have any change. So this is summarizing the level of infection again in these animals. So we've got the eyes in one column here and the urogenital tract, and these are the unvaccinated animals. So what this tells you is that a lot of the animals didn't change their level of infection. However, you can see that for 3 of them at least, their eyes increased, so they got worse, higher levels, 2 at the urogenital tract. So 5 of the animals had sites where the infection levels increased. Interestingly, 1 of them decreased, because that just tells you that there's a variation going on. Then if I give you the vaccinated animals down the bottom, and so hopefully you can be happily convinced again that in the second trial, in the vaccinated animals, none of them increased in their infection levels. Whereas if I remember you combined up here, this was 5. Whereas by comparison, the 7 that were infected decreased their infected levels. So this is hopefully a pretty convincing, again, second trial to show that we're able to use the vaccine to decrease the infection levels. The second type of vaccine that I mentioned that we're interested in is whether or not vaccine can reduce the progression to disease. So not just the infection level, but the progression to disease. So these are animals that were not diseased to begin with, but following the vaccination, some of them will develop disease over a 2-year period. So the data here in this particular one is that we're looking at new disease and shows that after vaccination, pretty dramatic. The small numbers, but nevertheless pretty dramatic. So in the non-vaccinated animals, a significant percentage that went on to get disease, but in the vaccinated animals, none of them did. And here it is again with the other adjuvant. And again, on the right-hand side here, I'll just mention to you that in the vaccinated at 6 or 12 months, many of them went on to get disease. At 6 months, none of them went on to get disease in the vaccinated group. However, some of them did go on eventually and get disease. And we're trying to work out why that would be the case. When we look at the mechanisms behind it, and I won't go into that into a lot of detail for you, but nevertheless, I'll give you some detail. So a bit like the flu vaccine, the influenza strains, there are multiple strains out there circulating in koalas. And so we asked the question, was it because in this particular case, the vaccine didn't quite match the strains that were out there? And the answer seemed to be no at that point. But I'll tell you something in a minute, that might not be the case. On this side, it tells us what part of the immune response we're interested in. So this is serology, so this is antibodies. We knew that there are circulating antibodies and they're important, but this is a disease that's at the mucosal surface. So it's at the mucosa of the eyes and the mucosa of the genital tract. So it's the IgA or mucosal antibody that's most important. And so what we saw here is in the animals that were perhaps not fully protected for the 2-year period, perhaps their levels of mucosal antibodies weren't as high. So that's something we're working on right now. So with that level of confidence, we then decided to do a study in, as I mentioned, animals that are already diseased. So this would be a difficult thing to do. And so this particular trial was done at a wildlife hospital. So these are still wild animals, but they're coming into the hospital because they've got either had a road accident or some dog attack, or they've already got chlamydial disease. So we waited till we could get enough animals that only had eye disease, ocular disease. In this particular case, we withheld the antibiotics. Normally they would give them some antibiotics in the eyes, pretty easy to do, but we withheld the antibiotics and we asked the question, could we use the vaccine on these animals and see if they would recover from it? This was a difficult study. It took us 2 years to do, and we got a total of 7 koalas that were suitable for this particular trial. So what we did was there were 3 grades of ocular infection. We didn't use the highest grade because we don't want animals to suffer, and ethically we didn't want to do anything that couldn't be controlled. So we used the lowest 2 grades, grade 1 and grade 2. So I'll give you an example here. Here's one of the koalas in the trial. So this is its left eye. And indeed, its left eye had a low-grade infection, even though you can't see it perhaps as easily from here. It was a grade 1 at the lowest level, and in the right eye, this animal here at a grade 2 at a score of 4.5. So we're going to see if the vaccine now given to these animals could not only reduce the infection level, but can it also reduce the clinical score. And so what I've summarized here for you is just the clinical score. So this is the clinical score. These are the 7 koalas I just talked about. And so what we have here is the clinical score going here, and these are the 2 eyes. So this is one eye and this is the other eye. So one eye actually didn't have much of a disease in it anyway, as I mentioned to you. And so this is the first 2 weeks. And not surprisingly, a vaccine is not magical. It takes at least 2 weeks for the immune response to kick in. So perhaps not surprisingly, the disease actually got in the eyes actually got a little bit worse. But then as the immune response from the vaccine kicked in, you can see hopefully a very strong clinical result. So the clinical disease went right down. Here's the second koala. Again, pretty good response. 3rd koala, 4th koala, 5th koala. And there's one koala here that didn't respond the way we wanted to. And I'm going to tell you why we think that's the case. But nevertheless, 6 out of the 7 koalas, no antibiotics, vaccine only, were able to basically cure themselves of their eye disease. And the reason we think that's the case is that this is a phylogenetic tree showing the different strains of chlamydia that are out there. The blue bars are the ones that were in the vaccine, where the vaccine came from. So you can see this one here matched up with the vaccine, this one matched up. We had one here, the animal that didn't respond ideally, didn't match one of the antigens we had in the vaccine. So the question is, how is that the case that it didn't have as good a response? So we think that's probably the case. And now we're looking at the possibility of matching the vaccine to the populations of koalas, because we've been doing lots of work to type the strains across the place. Just looking at the time here. So here's a nice photo to finish with. So it comes in at pre-vaccination, we withhold antibiotics, we just give it the vaccine, and hopefully I can convince you that it's recovered fully. And indeed the vets who are a tough group to convince at times, felt that the vaccine did such a good job that by the end of it, they were able to release the koala back into the wild without giving it any antibiotics, which means we don't use the antibiotics to destroy its gut microbiome. So that's a pretty successful story. Since then, we've tried to do a similar one with the urogenital tract, but it's a problem there because we can't withhold the antibiotics, because we can't know that the animal is getting better or worse as easily as we can with the eye. So we're still trying to refine how we can do that with the urogenital tract infections. So the pieces are coming together pretty well by here, and things are progressing pretty well. So I'll mention now, but then I'll go across to tell you about the koala retrovirus vaccine, and then at the end there'll be a couple more slides. We have 2 current large trials going on with our chlamydia vaccine, and one of these is in an area, not in Queensland, but south of Queensland and New South Wales, that has an even higher level of disease. So this will be an even higher challenge for the vaccine to get more data to see whether or not we're completely convinced. I will mention that what we are not going to do is we're not going to take healthy koalas, give them the vaccine, then purposely give them chlamydia. We feel that would be unethical to do. As I mentioned, the antibiotics are not going to be able to do ideally what we want. So we're relying on the fact of measuring koala responses in wild koalas, which therefore there are some limitations on that. But let me just switch for 10 minutes across to the development of the other disease that's only recently been discovered in koalas. It's a koala retrovirus. So the retrovirus is quite a complex thing, and I'm going to mention here that it's got 2 issues that we're worried about in koalas. One is to do with causing lymphomas and cancers. So just first of all, some background on the koala retrovirus. It was only first recognized back in 2000, so we haven't known about it for a while. Again, there are multiple subtypes recognized. It's quite complex in that it's, if I just go back to this diagram here to show you how the retroviruses work. So this is a retrovirus that attaches to the cell. It escapes the RNA, converts into DNA, and the first thing all retroviruses do is they insert their DNA into the genome of the host cell. So every single cell that gets infected with a retrovirus, part of the retrovirus DNA ends up in the cell. And then what happens, once that's in there, it then transcribes it, makes more RNA, more proteins, and it makes more virus associated. So that's pretty bad. That means every cell has got a copy of the virus in it. In some cases, in the northern Queensland part, the virus has infected gametes or germline cells. And when it does that, it ends up in the germline of the animal. And so that means that that animal will have that virus forever. So we know that some of them, KORV-A strains in New South Wales and Queensland, are endogenous. So every single koala is infected. Whereas down the south, only some of them are infected, and they're just infected at the exogenous level. So here's a diagram here just to show you that in the north, again, 100% of koalas are infected endogenously. As we get out of the south, some of them are infected, but some of them are not infected. And separately, we're trying to work out why how it's spreading. So as I mentioned, there are 2 basic reasons we don't like KORV from a koala point of view. It can cause lymphomas and leukemias, although we're still trying to understand the genetic mechanism of that. But as well as that, it looks like some of the strains of KORV actually make chlamydial infection worse. So it causes more chlamydial disease when they're co-infected with KORV. So this is the reason we're interested in making vaccines for both KORV and chlamydia. So I just put this one in just for those that are interested in phylogenetic trees. So this is where we think perhaps where KORV originally came from. This is the A strain, which is the most common, and then it's branched out with its slightly different genetic variants. So this is exactly what coronavirus, that's what the flu virus does. Yeah, it's what the coronavirus is doing right now. The differences are relatively minor, just small mutation differences until we end up suggesting that they're different strains in the case of KORV. So I'm just going to describe to you 2 vaccine trials we've done in the last couple of years with the KORV. And one of them is going to be in Adelaide and one is going to be in Brisbane. So I talked about, first of all, about the one in Adelaide. And the reason we did this one here is because, as I mentioned to you in that slide earlier, not all koalas in Adelaide are infected. In fact, the ones that are infected are exogenously infected. So we thought this would be the best place to start the vaccinations. Remembering, this is going to be a hard vaccine to make because KORV is in the genome of the koala. So it's actually even harder than a chlamydia vaccine. When I get to the Brisbane trial, these will be in endogenously infected koalas. This is a situation where, from the koala's perspective, it's got the virus inside its cells. So to make a vaccine against that is, some people would think it's almost impossible to do. So the first trial, as I said, was in South Australia. Small numbers of animals. We're restricted to small number of animals. That's all we do, and we vaccinate with an envelope protein, same as you would do in the influenza flu virus vaccines. So when we did that, we were very pleased with our results. So if I go to this side of here, these are animals that are KORV negative. So they're negative. When we vaccinate them, they make antibodies. These are antibodies we're measuring here, up to 50,000 units. A bit of a dip. We're still not sure why that's the case. So we can vaccinate koalas there. We can even take exogenously infected koalas. So if animals are KORV positive that otherwise don't make many antibodies on their own, and we vaccinate them, we're able to get them to make antibodies as well. So we're pretty pleased about that. We weren't sure to begin with that would be the case. More importantly, but, is what sort of antibodies are they? And we were able to look to see if they're neutralizing antibodies. So I'll use the example again from the flu virus. What's important for your flu vaccine, that it makes, when you get jabbed with it, that you make neutralizing antibodies. So in this case here, we're measuring at the beginning of vaccination and then after vaccination, did those antibodies neutralize the infection? Did they mop up or soak up the infection? And the answer is yes, we get neutralizing antibodies following vaccination trial. But this is a clincher experiment. So this is a question of a koala that's got retrovirus circulating in it. When it got vaccinated, the virus levels decreased over these 12-week periods. So after 8 weeks, they're already going down. But we couldn't get them down to 0 because it's a pretty hard thing to do. So this is pretty promising from that point of view. The second trial I'll just finish up with was done in Queensland. So this is harder again, because in Queensland, the retrovirus is actually endogenous. It's in every single cell. So here, we've vaccinated these animals. We're measuring the antibody response. So here's the pre-vaccination. There's what we think is low levels of antibodies. Well, once we vaccinate these animals, hopefully you can see that we're getting an increase in antibody levels in these koalas. This here is at 24 weeks. Since I put this graph together, we've actually got the 1 year data through, and it's going down a little bit, but after 1 year, we've still got these animals making antibodies to the retrovirus. We also tested for neutralizing antibodies, and again, we were able to get them to make neutralizing antibodies. I'm sorry that this graph is presented the wrong way around, but it's making more and more neutralizing antibodies. And finally, this is quite a complex graph here because we've got different KORV types. I've just put up KORV A and KORV B, but we tested for all of them. But nevertheless, the results with KORV A are very promising. So pre-vaccination, they have lots of virus circulating. After vaccination, we appear to have driven the virus right down to really low levels. Not as good a results with the other types of endovirus, but nevertheless, high levels to begin with, down and maybe up and down, bouncing around. We couldn't eliminate all this virus. That's because it's a more infectious virus. But nevertheless, these are very, very promising situations. So what we've ended up with the current situation is we've got 2 vaccines being developed, the chlamydia vaccine showing lots of promise, and I'll tell you in a minute, and then the KORV vaccine at a much earlier stage, but it's also showing lots of promise. So we've got 2 trials underway, or what we'll hope will be underway. One, again, is in this Moreton Bay region, southeast Queensland. What we're going to do here is we've tried a combination vaccine. So we've either tried chlamydia vaccine alone or chlamydia vaccine that's got KORV in it as well, to see if KORV, which is meant to cause some sort of immunosuppression and make chlamydia worse, if we vaccinate them together, is that possible? We're also hoping shortly to do this trial in the northern New South Wales area, but coronavirus, this was supposed to start at the end of last year. Coronavirus has stopped us in our tracks in February. But nevertheless, we're hoping to get it started at the end of this year. This population has very high levels of chlamydial infection and disease, so this will be a real test for the vaccine. I'll just finish with one last slide here, and before we can see if there's any questions you'd like to ask, or I can expand. So the question for us is, we'll keep doing research on this vaccine to try and understand it more, to work out what the mechanisms are, and to get better responses if at all possible. But at some point, we need to take this vaccine and use it, not really in a commercial sense, because there's not necessarily any money to be made here, but can we use the vaccine to vaccinate lots of koalas in Australia? And when people ask me which koalas will vaccinate, I have 2 or 3 answers for them. And one is, it would be logical to vaccinate all the koalas that come into koala hospitals around Australia. This is thousands of animals each year, unfortunately. So just vaccinate all of them. Also, when new roads are being built, when new shopping centres are being built, obviously then animals have to be looked after. And so those animals are coming into human contact, so to vaccinate those as well. And indeed, there are a number of other trials as well. And indeed, earlier this year, major bushfires in the south part of Australia, and lots of animals had to be rescued. So again, it would have been logical to vaccinate those animals. So the question is, how close are we to doing all that? Well, we're always doing larger field trials to validate, and these are underway. But the question for us right now is to decide how much proof we really need. As I mentioned earlier, we're not going to do this live challenge trial. That's just unethical. So we're at a stage now where we're trying to convince the government that we would be interested in rolling out these vaccines at some time soon, while we keep trying to improve it. And I use the analogy with our flu vaccine. So the flu vaccine is good, but it's far from perfect. I don't know whether most of you know that the flu vaccine, the immune response to the flu only lasts about 3 or 4 months in its strongest form. That's why you have to get vaccinated every year. And as well as that, of course, the vaccine has different strains of flu in it each year. So it's not perfect. So if we could do a similar sort of initial vaccine for koalas soon, and we can keep improving it as we go along. And we can then decide whether or not we need some sort of So I think just to finish up here, things are pretty promising. Hopefully we can use the vaccine as a management tool before too long, as well as seeing how it goes. I'll just mention now as well, a few weeks back, I was lucky enough to do a story that ended up in the New York Times, front page of the New York Times actually, where our koala vaccine work is actually being used as a model back for human vaccines. So there's no human vaccine for chlamydia yet. There is work in mice, but the question is, can a koala model, as clumsy as it might seem, be useful for a human vaccine? And the answer seems to be yes. So I'll just finish up by listing all the people. So lots of help from all different universities, councils, all our koala collaborators, Lone Pine Koala Sanctuary, lots and lots of people and councils and so on. And of course, lots of people, including students, both undergraduate students, postgraduate students, and researchers. So I'll stop there, and if there are any questions, I'd be more than happy to answer it. So thanks very much for your attention.
Thank you, Professor Timms, for that very informative talk. And I do have some questions. So, of course, do feel free to ask, if you're on the Zoom chat, do feel free to unmute and ask your question. Or if you prefer to ask it anonymously, just pop me a message in the chat. Of course, if you're watching on the live stream, feel free to answer questions through the live stream chat. But we do have 2 questions from our form that we sent out to our members before the talk. So the first one is, what would be the ecological significance of rolling out this vaccination program for koalas? And is there a precedent for vaccinating wild animals?
Okay, yeah, thanks for that. That's a really good question. Certainly the first half of it. This was a study done in southeast Queensland some time ago of a population that was in decline. So there was a series of populations. It's not one continuous population, because we keep building roads and what we don't do. There are a series of different populations. And there's populations, unfortunately, that are in decline. And some of them have declined to the state that they've become mini extinctions. So there's a study done in southeast Queensland of such of a population that had, the estimates were 10,000, number of years ago, 20 years ago, it had as many as 5,000 koalas, then 1,000 koalas, and the numbers are decreasing. So they did a study to try and ask, out of habitat destruction, wild dogs, road accidents, and disease, which was the most significant, and also which could you do to change? This was a modeling study. And so when that was done, it showed that even if you did all of those other things, including habitat, none of those could virtually turn that population from a decline around. Whereas if you came in with a koala vaccine, and even if that vaccine was only 60% effective, that would be enough to turn that population around from a decline to flattening out and then increasing. So that's a good example of how what ecological effect a vaccine could have. It doesn't need to be perfect, and it could take a population that's otherwise in decline and in effect rescue it. So that's sort of the approach. The other question was, is there other examples, I think, about other vaccines in wildlife? Well, there are. I don't have as many examples as I might for you right now. But nevertheless, we published a paper somewhat recently as well about this as well, saying that the model we've been using for our koala vaccine could be used for other wildlife situations as well. And I think the fact that we've done it against a bacterium, Chlamydia, as well as a virus, I think it shows that you can use vaccination in wildlife. As I mentioned, a challenge will be how to distribute the vaccine, but I'm sure there are thousands of koalas out there now that if we were able to use them in, as I said, in wildlife hospitals, wildlife care centres, when they're already captured, this would make a major effort. And in Australia right now, people are thinking even more about wild koalas that live on properties, sometimes shared with farmers. And in that particular case, rolling out a vaccine that way would still be useful initially. So hopefully that answers some of those questions.
Thank you very much. And perhaps that paper you mentioned, perhaps if you wouldn't mind sending one of our members the link to that paper, because I'm sure some of our members who are interested in, especially One Health now with COVID, has become far more popular interest, I think, amongst Cambridge students. So I'm sure that'd be of interest. And another question, again from that form, are there likely to be any negative health/ecological impacts for individual koalas or for populations that are associated with vaccinating them?
Yeah, that's a really good question as well. And we've taken a similar approach to what you would do with human vaccines from a safety point of view. So our early trials were done in captive koalas where there's no chlamydia, and we just delivered the vaccine to them. And we monitor them 24 hours, 48 hours, and longer periods of time. And indeed, before we can give that vaccine, we have to pass some pretty serious veterinary testing. So it has to be checked that it hasn't got any toxicity, and they analyze every component of it. And I can tell you, in the somewhat more than 200 koalas that we've now vaccinated, we've not seen a single adverse reaction from giving the vaccine per se. Of course, we are vaccinating wild koalas that already have disease. So sometimes our studies, some of the animals have disease, but we've not seen any situation. And it's a good question, but we don't want to give the vaccine including to diseased koalas and make them get the disease worse. We haven't seen that. The situation with the koala retrovirus is a bit more challenging, and we're being very cautious there at the moment because it's a retrovirus that gets into the germline, and so we have to make even more caution there. But the proteins we're using are purified proteins, and indeed we're moving now to producing them in a synthetic form so that there's no contamination or no other aspect there. The second question is a tougher one, and it's to do with a population. So some people debate about whether chlamydia is a natural control measure in koalas, but I can assure you that koalas suffer when they have chlamydial disease, both eyes and urogenital tract, and it makes them infertile. So I don't think we should ever consider that chlamydia is a natural control mechanism, and therefore treating populations to reduce their chlamydial infection levels, I think is the right thing to do. And we do know that over the last 20 years, the levels of infection and disease have got worse. And particularly now that things like heat stress, bushfires and so on are also challenging these animals. We're pretty confident that it's the right thing to go in and manage by administering a vaccine to help the koalas. I'm sure it's ethically the right thing to do, and I think from a population point of view as well. There's no suggestion that we could ever eliminate chlamydia. It's too clever a parasite, so we're not going to get populations where we eliminate the chlamydia and have problems for that reason. So I think we're just helping the koalas suffer less from what's a pretty nasty disease.
Thank you. And we have a question on the Zoom live stream chat. Yep. Has there been, or is there likely to be, are there likely to be ethical or economic complaints about, inverted commas, inverted quotes, interfering with wild species?
Yeah, good question. Yeah, I guess it's similar to the answer I just gave there, that I think, well, I think the best way to answer this, actually, that I haven't talked about at all, the people that we have a large project on, and the question is where did koalas even get their chlamydia from? It's a pretty tricky thing, but interestingly, there is reasonable evidence that koalas got their chlamydia from sheep and cattle. We know that sheep and cattle have chlamydia, and when we look at the strains of chlamydia that are in koalas, they are remarkably similar to that. So we know that in Australia, for instance, given that we've only been settled by white settlers for 250 years, that they brought their sheep and cattle with them, and they brought with them chlamydia. And so there's a possibility that what happened, at least to some degree, is that some of those strains of chlamydia jumped across into koalas. And then what we're seeing now is a little epidemic of that. So I think that we're still trying to prove that's the case, and it's hard to prove, and it might not be the case. The koalas might well have had their own chlamydia before that time, but there seems to be some commonalities. So if we contributed in any way to this infection in these animals, then I think we have every expectation to try and help through them and reduce it. I don't think the fact that we're using a vaccine to reduce chlamydial infections in a wild animal is the wrong thing to do. If that's the only thing they had against them, that would be fine. But we're also impacting them with reducing their habitat, unfortunately. We're building roads, and they get killed on roads, and we have, as I say, domestic animals. So if we're doing all these things to them, I feel that we have some right to, or expectation at least, to help them not suffer from this disease. So I don't think we're interfering with them. I would disagree that and anybody that's seen koalas with this disease would not for a minute suggest that we should leave them on their own. So I feel that we're not over interfering. We're providing something that's helpful. But a good question nevertheless. So thank you for that.
Thank you, and hopefully if that model there's turnout to that model with the 60% effective vaccine turning around the koala population decline turns out to be accurate, that'd be a wonderful thing, certainly. I now have a question, a personal question more about the vaccine itself. So would you mind elaborating on the adjuvants that you're using and how they work?
Yeah. Not easily for you. So can I tell you that one of them is a component that has 3 components in it, and this I'm somewhat limited to doing that. We collaborate with a group in Canada, VIDO, the Vaccine Against Infectious Diseases Organization, who are developing adjuvants, and some of that I guess is a little commercial, but I'll tell you roughly what they do. They develop these adjuvants for humans, but they also develop them for some wildlife there as well, including buffalo, bison, some strange animals where you wouldn't want to get too close to them. It has 3 basic components to it. It has one that's designed to stimulate antibody response. Second one is designed to, it's got a peptide in there that's designed to stimulate a Th1 response. In the case of chlamydia, we're after an interferon-gamma response. So we've been looking at that component pretty closely to see if that's we can we can enhance the interferon-gamma response, because that's what we want for an intracellular parasite. The other part is it produces micelles, so it's a lipid type component that produces micellar. So it delivers a component to the immune system that's likely to trigger dendritic cell uptake. Is the most I can tell you about there. So they're the three components to it. One to trigger Th1, one to trigger Th2, which is what we want in a chlamydia vaccine, and the other one is to compartmentalize it. We don't know how long it takes how long the vaccine stays at the location. I should mention to you, given that you've asked that question, that we did try 3 different delivery routes. And you would expect that the intranasal delivery route should immunologically be the best. And that's because we're trying to stimulate the mucosal immune system. We don't necessarily want circulating antibodies. We want antibodies and cellular response at the mucosal sites. And we know that delivering to the mucosa, so a nasal vaccine, should work best. We tried that in koalas, and we didn't actually see a better result immunologically. But the thing that convinced us is when we gave a nasal spray to koalas, they did not enjoy it at all. So we thought it would have been an easier way to deliver it, but it turned out not to be an easier way to deliver it. As I mentioned, we also tried oral delivery, and that's been done for chlamydial vaccines in mice in particular. And again, we didn't get a better result. And so it turns out that the subcutaneous delivery allows a little depot effect. So when it's given, it stays around for maybe a week or so. And it's also the method by which the veterinarians would prefer to give the vaccine. So it's both a practical sense, but it's also immunologically has turned out to be a satisfactory delivery route. We've given some koalas booster injections, and we don't think that's necessary, but we're trying to test that out at the moment. And I should mention that one of our earliest vaccines that was given was more than 8 years ago now. And we've actually been, that was in a captive animal. We've actually been able to get some samples from an animal 8 years later, and they're still producing antibodies to the vaccine. So I think that suggests that was reasonably good, and that hasn't had any boosting for the last 5 years at least. So that's probably the best information I can give you about the adjuvants, given that there's a little bit of commercial in confidence in there as well that I'm probably not allowed to give in great detail.
Thank you very much. That was very comprehensive. Of course, I understand that you can't share any commercial trade secrets. I think we might have one question from the live stream as well, from Yising.
Yeah.
Yes, thank you very much, Professor Timms, for your wonderful talk. I actually have one question. Because your research group conducts research on humans as well. So would you mind just elaborating your research on humans and probably compare it with your work on koalas? Thank you.
Yeah, okay, yeah, that's a pretty big question. So, yeah, so I was involved with mouse vaccine experiments early on, and then I've shifted my energy across to the koala ones. But what we're particularly interested in, what's really interesting from a human vaccine point of view, this is a small bacterium. It only has about 1,000 genes. And what we can't understand is how the cell tries to kill it, and that's why we're interested in the interferon-gamma. And an amazing story here is that the host cell tries to kill chlamydia by producing an interferon-gamma response. And interferon-gamma works in lots of ways, but one of the ways that it works is that it reduces the amino acid tryptophan. It literally starves the bacteria to death of tryptophan. Chlamydia are an amazingly clever organism. What they've done is they've got a couple of genes to convert the component indole back to tryptophan. So they basically rescue themselves by having just 2 genes that convert indole back into tryptophan. So the host is trying to deplete tryptophan, and the bacterium is putting tryptophan back in. The clever thing is where they get the tryptophan from. So in the female genital tract, so this is humans now, in the female genital tract, the microbiome that's there have indole producing bacteria. So amazingly, what these bacteria are doing in the genital tract is stopping the interferon-gamma tryptophan depletion system that we're trying to kill them with, and using the bacteria indole and converting it back. So this is an amazing adaptation that they're doing. What we found out now is that the bacteria, the chlamydia in the eye, are different, and they've actually turned those two genes off because there's different mechanisms going on there. And it's interesting, what we've been doing is comparing that in humans. We know in mice that they use a different mechanism. It looks like koalas use a different mechanism again. So from a broad chlamydia point of view, it looks like the different chlamydia strains that infect different hosts, so koalas, mice, humans, and the like, have all evolved a very clever way of overcoming the host attempt to kill them, which is why when we're trying to make these vaccines, which is the overlap with vaccine work here, we're trying to get an immune response that induces interferon gamma. So we want to swamp them and beat that response. So when I said to you, I think it'll be hard to make a vaccine that completely eliminates chlamydia. It's because they're very clever at overcoming everything we throw at them. Something as simple as indole tryptophan nexus there is a pretty clever thing that a bacterium with a very small number of genes has done to make sure that the host can't kill it. So I guess our approach is to do things that overlap both in animals, and that'll teach us things in humans, but also in the case of the koala, we can make a vaccine in this particular case or understand what it's doing. So it's been an amazing journey to see the different cleverness that different chlamydia have, and including, of course, in the human chlamydia. I should have mentioned as well that because different humans have different microbiomes in their genital tract, that also potentially explains why some people are more susceptible to getting chlamydia infections than others. And that's a fairly hot topic at the moment. We know that the microbiome affects HIV infection as well, because there are a lot of bacteria there producing a lot of important metabolites. So, you know, it just shows you the biological differences between humans or koalas and their susceptibility to these different infections that we perhaps wouldn't have understood if we weren't studying basic science.
Thank you very much for that answer. And it's also fascinating how these different infections can affect each other in terms of susceptibility. Of course, with the retrovirus and chlamydia. Perhaps, do we have more questions from the live stream or the Zoom chat? If not, I'll probably just squeeze one more question in for myself.
Yeah, sure.
All right, and then if there are any more questions from the Zoom chat, we'll ask one more and then probably we'll wrap up. So you mentioned that it would be very difficult to develop a vaccine for chlamydia-negative koalas, so that, well, in layman's terms, if I can term it, a preventative vaccine of sorts. Why would it be particularly difficult?
Yeah, I probably didn't explain that properly. It's probably not difficult to develop the vaccine. In fact, we'll give the vaccine to chlamydia-negative koalas, and I'm sure they'll make an immune response. It'll be probably impossible to stop when a koala gets infected, to stop it getting any level of infection, and that's the case with the flu vaccine as well. Now, you get a vaccine, but if you then come across the influenza virus, you wouldn't stop it cold. You would still get a very mild infection, but you would hardly notice it at all. The other question I meant to explain there is that it's very hard to test that vaccine. So what we did in those field trials is we took koalas out there. We took 30 koalas, and we didn't know what they were. So perhaps 10 of them were chlamydia negative, 10 of them had a low level of infection of chlamydia, and some of them already had chlamydial disease, and we gave them all the vaccine. So to test that vaccine in those chlamydia negative koalas, what we're relying on them to do is to go out and get infected, and given that this is a sexually transmitted disease, to get infected. And we know that the infection rate is around 10 to 20% per year. So what you're trying to do is test whether or not an animal that you've vaccinated, when it's getting naturally infected, and given that only 10 to 20% of them get infected, you're trying to see a difference there between your vaccinated group and your control group. So you need something like 100 koalas in each group to test that, because you're relying on only 10 or 20 of them to get infected and see if your vaccine works. So it's difficult to test. It's just scientifically difficult to do that to test a vaccine like that in a koala. What's done in human studies sometimes is the equivalent of a phase 4 study. People don't realize it's phase 1, 2, and 3, but there's also phase 4, and that is when you go out and then you, in the case of our situation, we would love to soon vaccinate a whole population of koalas. So this would be something like, maybe 100, 200 or even more koalas. So when you vaccinate a whole population, you can then start seeing this effect on chlamydia-negative koalas or indeed on herd immunity from a population point of view. People have been talking about herd immunity in the case of coronavirus. How many animals in a population or how many humans in an area need to be vaccinated to get herd immunity? So I think there's no doubt that vaccinating chlamydia-negative koalas, if we're able to vaccinate the majority of a population, will have a positive effect, primarily from herd immunity, if not from single animal immunity. And that's our next, if you like, our next study we'd like to do. Of course, that's a quite expensive undertaking to vaccinate a whole population of koalas. But these things do come up from time to time, so that would be interesting. That's a pretty good example as well as to how koala research can feed back to human research. It may be a bit easier to do some of these studies in animals and get the same sort of herd immunity levels that you hear people talking about. 60 to 70 to 80% is supposedly needed in the coronavirus situation in humans to get somewhere close to herd immunity. So hopefully we can do that sort of trial, perhaps in koalas with a different animal. So yeah, but it's a very good question. At the moment, we would give the vaccine to all animals. I should just mention what we haven't done yet, but is we haven't given it to very young koalas, again, from an ethics point of view. So animals that are still attached to their mother up to 9 months of age. But clearly that would be an important thing to do as well. But yeah, it's a very good question. Thanks.
Thank you very much. And hopefully perhaps with herd immunity in a koala population, of course, the young koalas would be protected as well. So
yeah, that's what we'd be hoping for. Yeah, I think we've got fairly promising evidence that we don't understand the transmission threshold, so we don't know what level of infection is needed to transmit. But again, if we can reduce that infection level down, either in individual koalas or then between koalas, we think that would be. We think we have every chance of success to do that. So that would still not remove chlamydia from the population, but it would keep it at a low enough level that that perhaps not many animals would progress to disease and not many would transmit. So that's our goal. Yeah.
So if there are no more questions on live stream, I think I'll wrap up here. So thank you once again, Professor Timms, for a fascinating and very informative talk on a topic that may at first sight not seem completely relevant to the current pandemic, but once we think about it further, about One Health issues, becomes more and more relevant to both the current COVID pandemic and to immunology and the prevention of disease in general, as well as to ecology and conservation. So thank you once again, and thank you to everyone who joined us and to everyone who watched us using YouTube, as well to people who will be watching the video later on. Just a reminder that we do have our varsity ski event coming up, so do sign up. You'll find more details on our Facebook page. Follow us on Facebook, Twitter, Instagram, or YouTube to keep up with our events. And we will have 2 more speaker events coming up in early September. One will be on neurobiology, and the other one will be, again, more related to the current pandemic on epidemiology and modeling. So once again, thank you so much, Professor Timms, for agreeing to speak to us. And thank you to everyone listening.
Thanks very much. I enjoyed it. Thank you very much.